All right, this is lesson 5.4, Life Science. Can cells use sugar as food? The standard we're working on is MSLS 1-7. Develop a model to describe how food is rearranged through chemical reactions, forming new molecules that support growth and or release energy as this matter moves through an organism. Our objective today is to investigate the use of sugar as food in cells, and we're going to do this using yeast. So in the picture, you can see some yeast here. We have seen that water and sugar can move into a cell, and sugar, which is food, can enter the cell because the sugar cells are small enough that they can pass through the semi-permeable cell membrane. So why do you think food needs to go into the cell? That is the question I want you to think about today. Have you guys heard of yeast? And do you know the role that yeast plays in food? Well, yeast is a single-celled organism. Yes, it is a living organism. And since it is too difficult to see what happens when sugar enters a human cell, we're going to investigate what happens when sugar enters a yeast cell. So remember, these are characteristics of living things. If something is living, it has to have the ability to grow. It has to be able to reproduce. It needs to use food for energy, and it gives off waste. All right, so we're going to watch this experiment, and as they do, I'm just going to kind of talk through what's going on. So I'm going to turn off their sound. So that we can just kind of talk about what's going on. So it's called What is Going On Inside of Me? And what we will do, we will conduct an investigation to see what yeast does with sugar. They give us a long time to read that, I know. Okay, all right, so here is the setup. They're going to use these test tubes. Each test tube is going to give 50 milliliters of water, and they're going to place a balloon. So this is test tube 1. All they put in test tube 1 was plain water, and then they just shook it up. Test tube 2 is getting sugar. That's sugar. And then it's also getting water. And they're going to put the balloon on top and shake it up. All right, test tube 3 is going to get water and yeast. So that is the yeast. They're going to put in water. And then he's going to put the balloon on top and shake it up. All right, test tube 4 is going to get yeast. That's the yeast. And sugar. And water. So he had the sugar and the yeast mixed together. And then shake it up. Okay. And so you can see the water obviously is just clear. The sugar has dissolved. This is after 5 minutes. We have just clear water, no change in our balloon there. This one was sugar and water, remember? The sugar has dissolved. You can't see the sugar anymore, but nothing else looks any different. This one was just the yeast and the water. You can see that the water looks kind of cloudy. The yeast is kind of clumped at the bottom down there. And this one had yeast and sugar with water. And we can see some little bubbles forming in there. And looks like the water is a murky white color. Okay, now this is after 15 minutes. There's still no change to the plain water. The balloon is not doing anything. No change in the sugar. Sugar water looks the same. Really no change in the yeast water, still looks white. And this is the sugar, yeast, and water, and you're going to start to see there's a little bit of gas in that balloon. Have a little bit of air in there. And we have quite a lot of bubbles. He's going to show you this up close in a minute. But there's quite a lot of bubbles in the top of that tube now. So they will zoom in on that and show you. There you go. Now you can see all those bubbles in that fourth test tube. There's a little tiny bit of bubbles in the third one, but there's a whole bunch of bubbles, and it has raised all the way up to the top of the tube. You can see. Okay, this is the next day. So after 24 hours, you can see all of the balloons are still deflated except for the one on test tube 4. The one on test tube 4, the balloon has risen and looks like it's filled up with some gas there. And when we look close up, now all those little bubbles are gone after 24 hours, and there's no gas produced in test tube 1, 2, or 3, but there was gas produced in test tube 4. All right, so here's a little chart that just kind of explains what happened as we went through. In test tube number 1, we saw really no change the entire time. The water stayed clear and there was no change to the balloon. In test tube 2, which had just sugar and water, the sugar dissolved, but nothing else really happened. There was no change. Test tube 3, we had just 1 gram of yeast and 10 milliliters of water. The water did look white. We didn't have any change to the balloon. We did get a little bit of bubbles right after 15 minutes, but then those went away and there was still no change to the balloon. There was not any gas in there filling up that balloon. In test tube 4, where we had sugar, yeast, and water, that's where we saw the changes. We saw the level of our water expanding. We saw bubbles in the tube. After 15 minutes, we saw lots of bubbles in that tube, and the balloon was getting a little bit of gas in it. And then after 24 hours, the water just looked white, but that balloon had expanded. And the bubbles kind of went away, but the balloon was expanded, so all the gas moved up into the balloon. So in the test tubes with water, yeast, and sugar, this is what was happening. There seemed to be more yeast, so it must have reproduced. Did you see how the level of the yeast kept rising on the test tube? So that yeast was reproducing. In order to reproduce, the organisms need energy. So the sugar supplied the cell with the energy it needed to grow and reproduce. The yeast produced a waste product, okay, and their waste product is called carbon dioxide. That's what you saw inflating the balloon. There was a chemical reaction taking place in the tube with the yeast, water, and sugar. The evidence of a chemical reaction were the bubbles that were forming in the test tube and the gas that was being produced and collected in the balloon. Gas is a waste product from the reaction. So no reaction took place in the test tubes without the sugar, and no reaction took place with just sugar and water. Therefore, there was a chemical reaction inside the yeast cell involving sugar that released energy for the yeast to grow and reproduce. Sugar can pass through a cell membrane. In this activity, yeast used sugar as food. So when the sugar dissolved in the water, the sugar molecules passed through. They were small and tiny, and they passed through into the yeast cells. And then the yeast cells use that sugar for food. So the sugar must be able to get into the yeast cells. In human bodies, cells use sugar for food through a chemical reaction that releases energy. So this is our fourth scientific principle. For the body to use food for energy and building materials, it must be broken down into molecules, which we learned about in the digestive system, and it has to be transported to the cells, which we're going to be talking about starting tomorrow. How does that sugar that's now broken down get transported to the cells? We know that once it gets in the cells, it's going to make energy. So how does it get to the cells? That's what we will be investigating tomorrow. Your task today is to complete Google Form 5.4. Please make sure you turn that in by midnight tonight so that I can mark you present for science today. I hope you enjoyed that little demonstration. You guys can even try that at home if you have like a water bottle and yeast and water. You can put sugar and yeast and water in your water bottle and put a balloon on there and shake it up and see if you can get that to happen as well. All right, I'll see you guys tomorrow. Bye.